Good afternoon, everybody. Today I want to talk about emerging viruses, which is a term that always bothered me, but since it's prevalent in the literature and in the world, we'll have to use it today. An emerging virus is the agent of a new or previously unrecognized infection. That's our definition. And this term emerged in the 1990s. But, in fact, emerging viruses are not new. They've always been emerging. We just realized it in the 1990s, so in typical fashion, we give it a name. And so that name is emerging viruses. But as you'll see by the end of this, they've been emerging since they existed. As far as we can tell, since the rise of agriculture about 11,000 years ago, new viruses have emerged, because the numbers of people who were in close proximity were enough to sustain the transmission of viruses among them. So before agriculture and commerce, most likely there weren't enough people concentrated in areas to sustain infections, and there wasn't the global travel, if you will, that allowed transmission to occur. So emerging viruses are sometimes an existing virus which has an expanded host range. We're going to look at some examples of that today. But most of the time, what's referred to as an emerging virus is a virus that's gone from some kind of an animal, either a wild animal or a domesticated animal, into people. And that's what we call a zoonosis, an infection of animals that can also go into people. Sometimes this leads to a new virus in the population. So we talked about how HIV moved from chimps to people in the early 1900s, and it became established there. So that's a zoonosis that is now a human infection. But often the infection is not sustained. And good examples of that are Ebola and Marburg, which jump from bats into people. They cause a very serious disease, but they aren't transmitted effectively within people. So there aren't global outbreaks of Marburg and Ebola. They're just isolated incidents of individual transmission accidents. And the public has its own perception of emerging viruses, of course, called killer viruses. The Ebola, of course, in particular, is pretty scary to people because it's highly lethal. But of course, it reflects a misunderstanding on the public's part, because these infections don't spread very much. So Newsweek and many other periodicals put it on their cover. My personal favorite is this book called Fever, which was published in the 1960s. And this is one of the reasons I got a PhD in virology. After I read this book, I wanted to work on viruses. And much of this story actually takes place at Columbia, which was at the time Columbia Presbyterian. And one of the early episodes of This Week in Virology, Dixon de Pommier was at Columbia when this happened, and he recounts his recollection of it. It's quite an interesting listen, but you can still find this book used on Amazon. I highly recommend it. It's really good. That, of course, was way before we knew anything about emerging viruses. There was not even the name coined for it. It was just a new infection. That book, by the way, represents or describes the outbreak of Lassa fever for the first time in Africa. These are 6 factors that drive the emergence of new virus infections. And they're drawn in this way to show how they overlap with each other, and they all contribute in the end, in the center here, to some way of new viruses coming out. And we're going to talk about a number of these today, not all of them. There are physical environmental factors, changing the environment by us, building a dam, for example. The microbe, the virus changing. Part of that is evolution, as we've talked about. There are ecological factors, interactions of wildlife with the environment. There are social, political, and economic factors, increasing population growth, government upheavals, poor populations that can't afford medicines. There are human and animal interactions that are new, that lead to transfer of infections. And there are also genetic and biological factors in both the virus and the host. So it's a pretty complex field of study, the viral emergence area. And it involves not just basic virology, most of which we'll talk about today, but also epidemiology, prevention, and response. Here's one of the overriding factors driving, or two of the overriding factors driving the emergence: incredible population growth and change that results in all the ecosystems as a result, by us occupying nearly every corner of the planet. And as you know, the population grows and grows, and it constantly goes into new areas. This is a photograph from National Geographic showing the lights in cities across the continent. And the whole world is basically like this, with little exception. You can see there are parts of South America not yet populated, but they will be. And as we move into new areas, that's one way that we encounter new virus infections. Other forces of disease emergence: globalization and rapid air travel plays a big role in carrying new infections around the globe once they're introduced into humans. Altered ecosystems, as we said before, changes in the environment. This is a pile of used tires. And I think we've mentioned before how the used tire trade has contributed to spreading mosquitoes around the globe. We have our pathogens, of course, which evolved. Don't forget that despite all the human-driven forces we're going to talk about today, the microbes are still evolving. Deforestation plays a big role in expanding populations as well. We'll try and give a few examples today of how these have led to new emerging viral diseases. The global aviation network is incredible. You don't see this very well, but these are all the airline routes around the globe. There are many more that are not showing up here. Basically, any place can be reached very quickly nowadays, and so it's very easy to bring a new virus anywhere in the world. And here's an example in South America, the North Amazon region of Brazil, which, as you know, is being encroached upon. We're deforesting it, but it's full of viruses. Here are just 183 arthropod-borne viruses that have been discovered in this area recently. And you can bet there are many, many more that haven't been discovered. And as we move into these areas and disturb the ecosystem, some of those end up infecting us. These are some of the viruses, the emerging virus infections that we talk about. They're not all of them. They include dengue virus, yes, which is an emerging infection. It seems like it's been around a long time. Ebola, of course. Hantaviruses, HIV, HTLV, another retrovirus infection. Influenza really is an emerging virus because it's emerging almost every year in a new form, and certainly every 20 or 30 years is a major pandemic form. And some of the forces that go behind that also contribute to the emergence of other viruses. Noroviruses we'll talk about later on, the agents of gastroenteritis. Sin nombre virus, and West Nile virus will be actually the topic of next Monday's lecture by Dixon de Pommier. So there are a number of ways that hosts and pathogens interact, and these form part of the way virus infections emerge, so we need to go over these, and these are shown here. There are 4 different kinds of host-parasite interactions. There's the stable interaction, shown here, where a host and a virus have evolved for a certain amount of time. It doesn't imply that nothing is changing. The virus is always changing, of course, as does the host, but it maintains the virus in the ecosystem in a predictable way. The evolving virus-host interaction is usually one where the pathogen has been introduced recently into the host, and both are still changing to reach a balance. So this typically involves the passage of virus to a naive population where it then multiplies and both the pathogen and the host evolve. There's a dead-end interaction. This can be fed either by stable interaction. So stable interaction could be a virus growing in a mouse in the forest. It's a stable interaction. It doesn't cause any disease in the mouse, but occasionally there are dead-end infections of other species. So it's a one-way route, and this is typically not spreading the infection to any others. And then we have the resistant host, where infection is blocked, doesn't really play a role in transmission or emergence in any way. So in stable host virus interactions, both participants survive and multiply. So many animals in the wild are infected with different viruses, whether they be insect or mammals, and both are quite fine. And that's a stable virus-host interaction. And some are permanent. There are a number of such interactions in people. Infections of humans with measles or herpes or human cytomegalovirus, smallpox virus, These have been going on for many, many years. They're stable, and as far as we can tell, they're permanent, although we've eradicated smallpox, of course, and maybe some of the others will change along the way. Sometimes more than one species is infected. Stable interaction doesn't mean there's just one species involved. So influenza infects many different hosts, and in many of those, it's a stable interaction. Flaviviruses and togaviruses as well. But this is not always the best way for a virus to persist. If it only infects people, for example, and people are wiped out, then the virus will go as well. We do see lots of examples of single host, stable virus-host interactions, but they have weak points, obviously. Now, the evolving host-virus relationship is characterized by instability and unpredictability. The virus and the host are trying to reach a common means or a level ground, if you will. Both are changing rapidly, and we can't really predict what's going to happen. The infection may be benign. It may not cause any symptoms or illness. It may cause death. Some examples: the introduction of smallpox and measles to natives of the Americas by Old World colonists. This led to huge changes in the native populations, which were naive with respect to these viruses. They'd never seen them before. They had no immunity whatsoever. Viruses were brought from Europe, and they caused terrible lethality in those populations. And the introduction of West Nile into the Western Hemisphere in 1999, which we'll hear about more on Monday. As these virus-host relationships evolve, the virus changes. It may become more virulent. It may become more transmissible. The host can change as well. If we look at long-term properties of a host, you can have wars, you can have famines or population changes, which also affect that evolution of that interaction. The dead-end interaction is often a consequence of cross-species infections, but not intraspecies. So human-to-human infections are not usually dead ends because the viruses that transmit among humans are adapted. But a Marburg virus going from a bat into a human is a cross-species infection, and that's a dead-end infection. Very often the host is killed very quickly, so there's no transmission. With Ebola and Marburg, the healthcare workers are typically infected. They're caring for the patient, but the infection rarely spreads outside the hospital setting. Sometimes the infected host doesn't transmit the infection well. So again, Marburg and Ebola don't transmit well unless you're in very close proximity to the infected individual. And these dead-end infections don't really contribute to the natural cycle of the virus in nature. They are dead ends. They don't contribute to the ecology or to the transmission of the virus in nature. So let's look at a couple of examples of these life cycles. Here's the life cycle of an arbovirus. Arbovirus is a name for arthropod-borne viruses, viruses transmitted among hosts by a variety of insects. And here is a virus, it could be one of many, that is transmitted among wild birds and domestic chickens in a cycle maintained by mosquitoes. So the birds are infected, and the mosquito bites the bird, transmits the virus to another bird, which then goes to another mosquito, which can then go to a chicken, and so forth. And this is a stable host-virus interaction. This occurs in the nature. The mosquito is fine. The wild birds and the chickens are fine. And so the virus reproduces nicely. Occasionally, there are accidental infections. Mosquitoes bite horses, in this case, or humans. They transmit the virus. The animals get sick. They may die. It's a one-way, dead-end interaction. The virus is not spread from human to human by mosquitoes, for example, or from horse to human or horse to horse by mosquitoes. So this is the natural cycle in nature. These are accidental hosts. Here's another example of viruses spread by ticks. So ticks bite rodents which have the virus. They pick up the virus, and they can spread it to other rodents. In some cases, the viruses are spread transovariously from mother to tick offspring, and then they can bite naive rodents. So this is a stable cycle, the rodent-tick cycle. And both the tick and the rodent, again, are fine, despite having virus multiplying in them. It's evolved over many years to that point. The ticks may bite people. You go walking in the woods or the high grass, you get a tick on you, it bites you, you get sick. It's a dead-end host. It's an accidental infection, and you don't transmit the virus to anyone else. People can also pick it up from goats, which get bitten by ticks. The ticks infect the goat. The person drinks goat milk, which isn't sterile, and picks up the virus infection. So they're both one-way interactions, dead-end hosts.
Is the goat also considered a dead-end host, or can the goat pass it on?
Yeah, the goat's a dead-end host as well. So not only humans can be dead-end hosts, the goat, the horse, and the people, right? Other kinds of dead-end infections, yellow fever in people is a dead-end infection. It's typically transmitted among monkeys and mosquitoes in the forest. When it goes to people, it's a dead-end infection. Same for dengue fever. It has a wild cycle between monkeys and mosquitoes. Marburg, Ebola, Hendra, Nipah, and the SARS progenitor all go from bats to people. One-way dead-end interactions. We'll talk about some of these. Lassa virus, the one that got me so interested in virology. Hunin, Sinombre are all spread from infected rodents to people. And again, the rodents are fine. They spread the virus among them, not by vectors, not by insect vectors, but by contact. Laboratory animal models for disease, yes?
I'm sorry, when you say animals are fine, do you mean if they're asymptomatic, or they should just live with the virus?
They don't get sick. As far as we can tell, they're not killed. They're fine. They multiply normally and breed without any consequence. Yeah. Whether they feel badly or not, I don't know the answer to that. Laboratory animal infections are really dead-end hosts. We have a model for polio in the lab. We infect with polio, gets poliomyelitis. That's a dead-end infection. That contributes nothing to the cycle of the infection in nature. So just about every laboratory infection, for the most part, is a dead-end infection. And they typically don't even transmit it to each other, with few exceptions. So when we talk about emerging infections, there are really two main steps involved. There's the introduction of the virus into a new population. We'll talk a lot about how that happens. And then the virus becomes established in the population and then is disseminated. So the dissemination occurs nowadays because of our transport systems globally. And we'll see some examples of that as well. Most of the encounters that we have with new viruses don't lead to anything. So chance encounters are never detected. If you go down to the Amazon rainforest, you'll probably be infected with some virus, and likely you won't ever get ill, or you wouldn't even know that you were infected. And from what we've learned in this course, you know that your innate system probably deals with the infection, and that's the end of it. And you'll not transmit it to anyone else. It doesn't multiply sufficiently in you to transmit it. It never is secreted in any effective way to be transmitted. So there are many reasons why most encounters with viruses never go anywhere. And a lot of what you've learned in this course will let you explain that. In order for an infection to emerge, the virus has to have access to susceptible and permissive cells. Remember, the ones with receptors and the ones in which the virus can multiply. And the virus needs to multiply and then spread from that initial host in a population. So population density is important. You have to have enough people around for the virus to spread. If the health is poor, that will encourage the spread of an agent within a population. In the end, what is really important is that you have to have serial infections, a chain of transmission. If a virus is going to be established in a population, it has to be able to go from one host to another. So starting from that initial individual, it has to multiply effectively and spread to others in a population. And I think you'll see a lot of the characteristics of that in the specific examples that we talk about. So we are always providing new ways to encounter viruses. These are just some of them. Air travel, of course, is a big one. We build a dam. We change the ecology of an area, and species interact in different ways. Irrigation, rerouting wildlife migration patterns, wildlife parks where animals are confined to areas and people interact with them. Hot tubs, air conditioning, blood transfusion, they're obvious. Xenotransplants, putting animal tissues into people, good opportunity to introduce new viruses. Moving livestock and birds all over the world is a great way to move viruses that infect them. The way we look at drug abuse and sex has a big role in spreading infections. massive deforestation, the used tire trade, as we talked about, population growth, and of course, daycare centers is a big one, which spreads infections all the time. So here are some specific examples of parameters that facilitate transmission of viruses to new hosts. So if the virus requires contact with bodily fluids to be transmitted, hunting, predation, or eating wild game can do that. You go to an animal zoo, or you have a brand new pet in the home which has a virus. Hamsters often bring in lymphocytic choriomeningitis virus into the home and they infect people. Another example is sharing of a resource with a different species. We'll see this in the case of fruit bats and pigs and the origin of Hendra and Nipah viruses. Sharing, sometimes rodent or insect vectors are shared, and that's the case for Japanese encephalitis virus, which is spread by a single mosquito that feeds on multiple hosts. And often species encroach in the habitat of another. We go into the jungle and we're bitten by a mosquito with yellow fever virus in it. If we hadn't gone in the jungle, we wouldn't get bitten. So that's another way that we bring the infection back. So the first couple of examples are viruses that have spread from fruit bats, which are also known as flying foxes. They're present in many parts of the world. Very large bats, wingspan about this big. It's not these little guys that twitter around in the night. They're this big. They're very scary. And they, from Australia and Southeast Asia, 4 unknown paramyxoviruses have been identified from these animals since 1995. And that includes Nipah and Hendra, which we're going to talk about. And these cause very severe disease in animals and in people. And a paramyxovirus, you may remember, is an envelope virus. Measles virus is a paramyxovirus. An RNA genome inside, glycoproteins on the surface. Standard paramyxovirus. So Hendra and Nipah both look like this. The Nipah virus emerged first in Malaysia in 1998. There was an outbreak of both respiratory and neurological disease on pig farms there. As a result of this first outbreak, there were 100 human deaths, and they decided to kill 1 million pigs to stop the transmission of infection. So this is a common occurrence when emerging infections jump into the food animal chain. You kill millions and millions of animals. The way this virus spread to both pigs and people, so both the pigs got sick and the people got sick, is through fruit bats. So fruit bats are full of Nipah virus. They excrete it in the urine, but they're fine. The fruit bats are healthy, and even though they have ongoing replication continuously, they are absolutely fine. And what happens here is that you build a pig farm, and then the farmers, for some reason, plant mangoes right next to the pig farm. The flying foxes like to eat mangoes, so they fly over to the pig farm. So maybe their habitat is being encroached upon or being destroyed, or the pig farm is close to where they live. They come and eat the mangoes, and then they get virus on the mango while they eat it. And they leave some bits of mango on the ground. The pigs eat the mango, and they get infected. So that's how the virus gets from the bat into the pig. So this is an unnatural route of transmission. Normally the domestic pig and the fruit bat would never see each other, but you build a farm near the forest and this is what happens. Of course, people take care of pigs on pig farms, so the virus goes from the pig to the human and the humans get sick. Yeah. Yeah, they do. Yeah, they have a typical mammalian immune system, yeah. And, but this virus is able to multiply in them without a problem, so it's probably evading it in some way. But the bat is fine. It's not ill, yeah.
So do humans not get infected by eating mangoes, or is that just not a secondary source of infection?
No, there was no incidence of human eating the mangoes here. Yeah, the transmission was always from the pig to the person. I mean, if you did, you would probably get it, but. You want to eat a mango lying on the ground covered with bat urine. Right, so mangoes in the trees are okay because the bats haven't gotten those yet.
So only when the mangoes fall to the ground?
So the bats take the mangoes and they rip them off and they eat them, and then they leave it on the ground and what's left, and they've urinated on them at the same time. So don't eat those mangoes lying on the ground. That's the moral of the story. Hendra also came from flying foxes. This is a virus discovered first in Australia in 1994. There was an outbreak at a racehorse training facility. So in this one, 14 racehorses died, and then the guys who were taking care of them, some of them died as well because they're getting infected from the horse. In this case, it again spread from flying foxes to the horses. We don't know the connection here yet. There's no mango connection with the horses and the fruit bats, but there's some other way, probably through the urine of the flying fox or some other way. They go into the horse stall and contaminate it. And this virus has spread from its initial place in Brisbane, in the suburb of Brisbane, all around the northern coast and the eastern coast of Australia, and also spread to Southeast Asia. So the Hendra location now is in red here. So it's moving into Southeast Asia. And this is the Nipah location in blue. These are all the areas where there have been outbreaks. So from the original one in Malaysia, it's spread to most of Southeast Asia and India as well. So that's one example of how you encroach upon the natural habitat of an animal, in this case the bat. You build racing facilities or pig farms near forests where the bat lives, and you have these encounters which eventually lead. Now, to infection, it's not clear that Hendra or Nipah are yet being transmitted among humans. These are all dead-end infections, but the possibility, of course, remains that they might evolve to be transmitted. So we have to keep a very close eye on this. Another example is yellow fever virus. So many, many years ago, yellow fever was an infection in Africa between mosquitoes and monkeys, and both, again, were fine. The mosquitoes were fine, the monkeys were fine. When trade began to come from Africa to the New World, we brought the virus with us, and it was introduced into the New World, and the monkeys there, New World monkeys, got sick because they weren't used to the virus. They hadn't adapted to it. And of course, the humans were also infected as well, and they brought it to the New World. So this was brought by colonizers and traders to the New World, and this is the graph of an outbreak in Philadelphia in 1793. The virus apparently came from Santo Domingo. So it's brought in by a mosquito or an infected human, and then local mosquitoes pick up the virus and spread it. And you can see this outbreak peaked in October or so, 10,000 cases, 5,000 deaths. And then when the temperature started to drop and the mosquitoes die, then the outbreak goes away. This was a big problem in Eastern American cities at this time. The disease was imported from the tropical areas, and everybody was very frustrated because they didn't know how to control it. Of course, they didn't know it was a virus. They just knew it was a disease. And it wasn't until 1900 that the virus was identified and a vaccine made in 1937. But even with this vaccine today, there's still 10,000 deaths just in South America from yellow fever. Diseases of exploration and colonization we've talked about a little bit, but in general, when a virus is introduced into a naive population, you have explosive spread, epidemic spread, and a lot of disease. And this is what happened when colonizers moved from the old to the New World. So smallpox, which arose in the Far East many, many years ago, maybe from a gerbil infecting a human, moved to Europe, where it just spread like crazy and everybody got smallpox. And then it was brought to the New World by the colonizers. And this totally changed the balance of human populations there. The Spanish colonizers brought it to the Aztecs, for example, via Hispaniola. 3.5 million killed by smallpox alone in 1520. And that's why Cortez was able to conquest these people with very few soldiers. So there are many examples of this. The virus is brought over, and this population is naive. There wasn't global travel back then. Remember, the colonizers, the explorers, were the first ones to do this travel. They brought infections with them, and they ended up changing the balance of populations. Another example is how a change in the human population and the environment leads to emergence of a disease. So I've told you before that polio has been around since the Egyptian times. We think we have some records of it from 1500 BC. But it was always a sporadic disease, a few cases here, a few cases there. It didn't emerge as an epidemic disease that is occurring in big outbreaks of thousands and thousands of people until the 20th century. And that's shown in this slide here. This is just a charting of polio cases from 1885 to just past 1915. So very little in the late 1800s, and then at about the turn of the century, you see outbreaks of increasing number. So why is this? Well, this is called the disease of modern sanitation. Before this time, the virus spread quite freely among people, and at a young age, as soon as a baby was born, it was typically infected because sanitation wasn't very good. There's nothing as a sewer, no such thing as a sewer. So it was very easy for this fecal orally transmitted virus to spread in the population. So babies were infected early on. They became immune. Some of them developed polio, but for the most part, there weren't epidemics because everyone was infected at a very young age. Now, when you improve sanitation, you separate the contamination of a newborn from feces for many years. You now build up pools of susceptible individuals, and when virus is introduced into them, you have an epidemic. So this is a very interesting way that our changing sanitation actually produced an epidemic disease, and we call this the disease of modern sanitation. Changing climate and animal populations also contributes to the emergence of infections. So this is the story of the hantavirus, which was first noted as causing this hantavirus pulmonary syndrome in the Four Corners area of New Mexico in 1993. The Four Corners is where 4 states join exactly. It's a little marker in the ground. If you go there, it has a little two lines showing where the 4 states come together. And in this area, there were outbreaks of very unusual and severe pulmonary disease in 1993. It turned out to be caused by a virus, which is now known as sin nombre virus. And that virus came from the deer mouse, Paromyscus maniculatus. And this took a lot of detective work to figure out. They isolated the virus from some of these sick individuals, and then they went into the region and found the virus in the mice as well. And before we continue with that, this was originally called Muerto Canyon virus because that's where the first isolate was obtained. There was a canyon near the Four Corners called that. But the people who lived in Muerto Canyon said, we don't want this virus named after us. So the CDC called it sin nombre instead, which of course means no name virus. Right? So that's a good story. So hantaviruses are enveloped viruses. They're part of the bunyavirus family, which we haven't talked about very much. But these are typical envelope viruses. They have glycoproteins on the envelope. They have an RNA genome, which consists, in these cases, of 3 different RNAs. And so it's sort of like the influenza genome that's segmented, but there are 3 pieces. And these are wrapped in protein, as you might expect. Now, the virus jumped from mice into people because of a change in the climate and the animal population. This is a Paromyscus maniculatus mice here. And these mice like to eat pinon nuts, among other things. And it turns out that in 1992-93 was more rainfall than usual, and the crop of pinon nuts was bigger than it had ever been. And people harvest these pinon nuts, and they store it in their houses because they like to eat them. And the mice like to eat them as well. So the idea was that the mice came into the houses to eat the pinon nuts, and there they shed the virus in the urine and made an aerosol basically in someone's home, and that transmitted the infection to people. So an unusual year, a big crop of pinon nuts, that helped the mouse population to rise as well. And remember, these mice are all infected with these hantaviruses. They're fine. They don't have any problem. They shed it in their urine. They spread it from mouse to mouse via the urine. But when they go into someone's house and put the virus there, then you get infected. So the virus is in the droppings, and contaminated blankets or dust from floors provided opportunities for human infection. So the Epidemic Intelligence Service went into houses where cases had occurred, and they took samples, and they found the virus in dust on the floors and in blankets. Now, this is a rather rare disease because it's not spread from person to person. We are dead-end hosts. It comes from the mouse into us. We get very sick. Many people die, but we don't spread it to other people. So it's a rather rare disease. It hasn't adapted to us to be able to spread. This is a map of the distribution of Paramiscus maniculatus in the U.S. The red dots, the green area here, the shadow is the range of this mouse. So it's not existing just in the Four Corners area. It's in a large part of the United States. And that is where most of the isolations of sin nombre virus have been made. Those are the red dots. So it's pretty much within the area of the mouse. So this virus is endemic in the Paramiscus maniculatus species, and that is the virus that causes these cases of hantavirus pulmonary syndrome. But you can see from the number that as of 2003, it's not a very frequent infection, 350 cases. But when it does occur, it can be fatal. So this infection, which is a zoonosis, right? It's normally a mouse infection, but spreads to people. Potential diseases lurk if we intrude upon another host-parasite relationship. So it's the virus-mouse relationship that humans intruded upon by being in the same area. Often this is inescapable, of course, but it's an example of what can happen. The next example is SARS. And this is an email that was the first indication that something was going on in China. There's a very interesting website called promedmail.org, which is run by the International Society for Infectious Diseases. It's basically an alert system. When things happen globally, people send notifications to ProMedMail. And this is an email from February 10th, 2003. From this fellow, this morning I received this email and found nothing pertained to it. Does anything know about this? Have you heard of an epidemic in Guangzhou? An acquaintance of mine from a teacher's chat room lives there and reports that the hospitals have been closed and people are dying. So this was the first indication in the U.S. that something was going on. And remember, this is now an electronic era, so it would have taken a long time in previous years for us to get this information. So this turned out to be a new infection called SARS, and it was basically an outbreak of severe atypical pneumonia in this province in China in November of 2002. There were 300 cases and 5 deaths. The disease had a short incubation period, beginning with fever and typical symptoms of an infection, moving on to dry cough and shortness of breath, and then many people requiring mechanical ventilation support. So it was called severe acute respiratory syndrome. One of the physicians who was treating the first patients in China went to Hong Kong in February and stayed in this hotel, the famous Metropole Hotel. You search for that, you'll get all articles on SARS. So this is a very fancy hotel where people from all over the world stayed. So he stayed on the 9th floor. He got sick. He died the next day. They took him away. And then everybody else on the floor went to other countries, and they didn't know they were infected. So 10 people flew to these countries. They didn't have symptoms yet. They flew to the countries and brought the virus there. And this has been very nicely mapped out. It ended up spreading to 8,000 people in 29 countries, starting with this physician who left China and infected all these people. And this has a 10% mortality associated with it, so it's not an insignificant disease. So the virus is a coronavirus, which we haven't talked about much. These are enveloped viruses with RNA genomes. They're classified into 3 antigenic groups. And I want to show you this simply to show that there are human coronaviruses. They were known before SARS was identified. There are also coronaviruses of a variety of animals. And here's SARS coronavirus, which is in antigenic group number 2. These are enveloped viruses in the EM. They look like typical enveloped viruses with the spike glycoproteins being very prominent. This is a schematic on the right of the virion. It's about 120 nanometers in diameter. As you would expect, it has surface glycoproteins, which are involved in receptor attachment and membrane fusion. There's an RNA genome in the center, which is plus-stranded. And it's the longest. The coronaviruses have the longest RNA genomes, over 20 kilobases in length. Before SARS, there were other coronaviruses of humans that were known. These caused a variety of respiratory diseases, but none as severe as the SARS coronavirus, which was called atypical pneumonia, unusually severe pneumonia. These viruses are transmitted by respiratory secretions. Mainly adults and healthcare workers were at risk for SARS infection, as you might expect. The virus spread everywhere in the end, and there were no vaccines or antivirals available against it. So the SARS coronavirus is transmitted by respiratory droplets. It enters your tract, multiplies in the epithelium, very much like influenza or rhinoviruses, eventually makes its way down to the lung, where you have very severe lung injury. And we're not sure why that is so, if it's a consequence of virus multiplication or the immune response or both. After that initial outbreak in China, the WHO called for a global alert. They called this virus SARS at that time. There was a second alert on March 15th after the outbreak from Hong Kong. And at this point, they said, here's the case definition. Be careful traveling. Be aware that this disease might be present. Unfortunately, travel continued after that first announcement, and that was spreading the disease to other countries. And by the end of March, WHO was saying to airlines, look, you have to watch for people who might be infected and try and restrict international travel, otherwise we're not going to contain this virus. So this was the outbreak in Hong Kong. You see it peaked in about at the end of March here, and then tapered off by the summertime. So 753 cases, 1,753 cases in Hong Kong. The impact on the economy was substantial. Here's a very nice poster from Hong Kong. Hong Kong will take your breath away, which we all like to show. This was circulated just before SARS outbreak, and indeed it will. The airports in Hong Kong were empty, which is very unusual because travel was being restricted. In the end, the global cases, which you can't see here, about 8,000 global cases, mainly North America, some in Africa, and Asia and Australia. Oh, there we go. So 8,000 total cases, 774 deaths for 10% fatality rate, US and Canada, Europe, and Asia, as well as Australia. So it did spread globally. And a big part of stopping the spread of this infection was informing the public health people what to do about it and how to find it. So for example, in airports, there were these temperature sensors installed which could tell if someone had a fever. And in Hong Kong in particular, if you had a fever, you were not allowed to travel. So if you were detected with a fever here, you were contained. Many posters were made and distributed describing atypical pneumonia, what it was and how you could prevent it in various levels for different populations. And this played a big role in the public health response and keeping the infection checked. There was a lot of surveillance and early warning, a lot of case investigation and tracking, trying to find out where the virus came from. We had no vaccine, we had no antiviral agents. This infection was contained largely through travel restrictions, quarantines, and public education. Very interesting example of this. So a lot of infection control education for both healthcare providers in hospitals taking care of these patients. So a lot of detective work was done to figure out where the virus came from. And here's the origin, probably from the meat market in Guangdong. So human sera collected before the outbreak in Hong Kong don't have antibodies to the virus. So it looked like the virus was brand new. The early SARS cases in Guangdong were in handlers of animals for the exotic food market. So there is an open meat market there where animals are brought in from the countryside, they're slaughtered and then sold for food. And many of these individuals were among the earliest cases of infection. In fact, the animal traders had higher prevalence of antibodies than control groups. We'll look at that data in a moment. They were able to track back the outbreak to an earlier time point than Guangdong. And in fact, as early as November 2002, they were able to recognize a case elsewhere. And then they subsequently recorded smaller outbreaks nearby, 11 cases here, where the index patient, that is the first patient infected, was a cook in a restaurant. So he probably got infected from the meat. Another outbreak in December, and then continuing towards the first outbreak in 2003. So a lot of work was done to establish the etiology. These are results of serosurvey tests, antibodies to coronaviruses in humans in Guangdong. And these are the various occupations that were looked at, the number of people and the percent that had antibodies against the SARS virus. So you can see the wild animal traders, the people who capture the animals and bring them into the market, had the highest seroprevalence, 40%. Animal slaughterers had high as well, vegetable handlers a bit less, and the control group, which were hospital workers who did not work with patients with respiratory disease, they had no antibody, and there were 60 of them looked at. So the origin was initially thought to be this animal called the palm civet, Paguma larvata. The virus was initially isolated from these animals, which are brought to the market from the countryside. They're slaughtered and people buy them for food. So in May, the virus was isolated from palm civets. And there was some evidence mounting that the infections were transmitted from civets to humans. So civets were all slaughtered. They weren't brought into the market anymore, and quarantine was imposed. But it turned out that these were probably not the reservoir. They probably passed the virus on to people. But the viruses isolated from many different civets were all very similar, and the virus was often not found in civets either wild or on farms. So this suggested introduction of the virus from another source. And so further work showed that bats in the countryside in different areas of China had SARS-like coronaviruses in them. And these were isolated in provinces that were very far apart. So these are probably the reservoirs of the virus or the precursors. As you asked before about the immune system, 84% of these bats had antibodies against SARS coronavirus. They're fine. Remember, the bats are fine. They're infected with the SARS corona. They're making antibodies, but they're still making a lot of virus. And these viruses were very similar to the viruses isolated from people. So that was probably the source of the outbreak. So the hypothesis is that the bats are the reservoir. They're transmitted to civets, which then are where people pick up the infection in the market. How did the virus adapt to us? Well, they sequenced many, many viral genomes from animals during and after the epidemic, and to try and answer the question, which changes lead to adaptation? It turns out that the spike glycoprotein in the envelope, which attaches to the receptor, is a major player in the adaptation of the virus from bats and civets to people. Human and civet viruses differ by 4 amino acids in this spike glycoprotein. But the human and civet strains have a very different affinity for the receptor for the virus, which turned out to be human angiotensin converting enzyme 2. So this is a surface protein on cells. This is a receptor for the virus. The human receptor binds very well to the human SARS isolates, but very poorly to the civet isolates. So in going from civets to people, the variants were selected that could bind better to the human receptor. And here's a model of how that is believed to happen. So this is a model of the receptor on top binding to the viral glycoprotein on the bottom in yellow. So the glycoprotein interacts with the receptor, and the human SARS spike interacts very well with the receptor ACE2. The civet SARS virus has 2 amino acid differences from the human, which make it a very poor interactor with the human SARS receptor. So the civet strain doesn't multiply well in people because it doesn't bind the receptor well as a consequence of these amino acid changes. There was an isolate of human SARS taken later in 2003, 2004. This was a less mild version of the infection. It didn't cause as much lethality. And this had an amino acid change here, which reduces the interaction with the human receptor. So you see the virus evolved from a poor interactor in civets to a very good interactor in the initial human isolates with the human receptor, and then backed off a bit, if you will. So it looks like the virus receptor interaction evolved so that the virus could multiply better in people. And really what happened was, remember, the virus in civets is a quasi-species, and the quasi-species probably contains some viruses that just randomly could bind the human receptor really well. When that virus went from a civet into people, those variants outgrew everything else, and those are the ones that caused the epidemic. So SARS, yes? What about the bats? Civet viruses are very similar. So they bind poorly to the human receptor.
Are you suggesting that the original SARS came from the civets that also?
The original SARS was in bats. The reservoir is bats. It then went into civets. The civets were brought into the meat market, and then it got into people that way. So will it come back? Because right now there's no SARS globally. There's no transmission at the moment. The most recent cases were laboratory acquired in April of 2004. And I told you about this less severe isolate, which appeared in December, January 2004. This was probably from a separate civet to human transmission from the original. It had enough differences that it could be identified as a separate transmission. So in other words, if you keep bringing civets to the meat market, eventually you're going to bring another virus like this back in. Interestingly, these viruses that infect people, both this strain and the earlier, more lethal one, have a 29 nucleotide deletion in one of the accessory genes of the virus. And this is not present in civet or bat isolates. So somehow this may make the virus more virulent in people. Yeah.
Is the less lethal strain more antigenically compatible with the?
Yes. It just has these two differences in the receptor binding site that make it bind less well. And it's probably a big reason why it's less lethal.
So we could make a vaccine?
You could, if it were stable. Yeah. But unless you wanted to make an inactivated vaccine, then you could do that. These viruses are still in animals. They're in bats, and they are in exotic animals that are eaten in China. They are isolated regularly. So not just civets, but they come into other animals as well, because there's contact between the bat and these animals in the wild. So probably at some point there will be a reintroduction, but we're ready. We're more ready now, so it probably will be easier to recognize and contain the infection. So this is what the outbreak looked like. The SARS outbreak is in the red bars, and you see it occurred just at the end of the influenza season. So it actually fooled a number of people into thinking it was just the tail end of influenza, but it turned out to be a distinct agent, of course. The impact is very interesting. Before SARS, there were 12 coronaviruses and maybe 12 people in the world that worked on these viruses in the laboratory. But of course, when this happened, everyone got lots of money to do whatever they want. We now have 36 coronaviruses because there's all kinds of surveillance going on for coronaviruses in animals. And there's been over 4,000 publications since the SARS epidemic on coronaviruses. So looking for viruses in bats is now a big business. I know a number of people who are out there catching bats and sequencing the viruses, and they find SARS-like coronaviruses in them all the time. Viruses also emerge as our technology improves and allows us to detect them. So there are many reasons why viruses emerge, but sometimes we don't pick them up unless we have good technology. And a great example are the caliciviruses, the plus-strand RNA viruses that cause gastroenteritis. 23 million cases a year of gastroenteritis in the US. These viruses can't be cultured in cells, so the only way we could discover them was when we had a technology that didn't depend on growing the virus. So these were discovered by DNA-based methods. Where do we get these infections? Water and food sources that are contaminated, aquatic theme parks, and cruise ships. So it turns out these are among the viruses that are excreted by whales. Remember in the very beginning, whales excrete 10 to the 13th caliciviruses a day in their feces. And some of these viruses infect us. They infect other marine mammals and they're spread to us. So often at aquatic theme parks where the big whale is jumping out of the water and you sit in the front row because you want to be splashed, well, don't do it next time because you can get gastroenteritis. And so you get a lot of outbreaks from that. And then people get infected And maybe they work on a cruise ship, so they go to work and then they handle food and then everyone on the cruise ship gets infected. So that's where we think these viruses come from. They're marine viruses that are infecting us, and they also infect an enormous number of terrestrial mammals. And they're also, of course, in shellfish. Shellfish, of course, just filter the ocean water and concentrate whatever's in it. So if you like to eat your raw shellfish, you might get gastroenteritis caused by one of these viruses. The good news is that after a few days you're fine, but it's not very pleasant. So that's just one example of a virus that we only discovered as technology allowed us to do so, and now it turns out to be incredibly important. Now, don't forget, we're talking about a lot of ways that we impinge on nature and get infected, but don't forget evolution is always taking place here. The virus is evolving at incredible rates. Remember, the quasi-species were evolving as well. So this evolution is why we have such a diverse virome in nature. It allows the viruses to adapt to new hosts. So SARS could adapt to humans because the humans selected out one of the quasi-species that was present. And this also goes for antimicrobial resistance as well. Remember, evolution, mutagenesis leads to drug resistance. So here's an example of hantaviruses in New World rodents. These are just some of the ones that we know about. We talked today about C. nombre, which is in Peromyscus maniculatus. But there are other similar deer mice that have similar hantaviruses in them. And one of them caused a case of hantavirus pulmonary syndrome out on Long Island a number of years ago. A different mouse, a slightly different virus. There are also other kinds of animals here. There are rats, cotton rats, and these are all this name of the animal species, and the name of the virus that's been isolated from it is on top. So you can see these are everywhere. The orange ones are already known to cause human disease. So there's a lot out there. There's a lot of potential for new emerging viruses to infect people as we disturb habitats in the ways that we've talked about. Here's a really example of how selection, another example of how selection leads to emergence of a new pathogen. For those of you with pets, this is canine parvovirus. So nowadays, if you have a dog, you should get it immunized against canine parvovirus. But before 1978, this wasn't an issue. And this is a virus that arose from cat to dog transfer, and it took just 2 mutations to do this. So this virus was identified just in 1978 as a new disease agent in dogs that caused gastroenteritis and heart infections. So that it can be lethal, and that's why you get your dogs or cats even immunized against it. It evolved from a cat virus called feline panleukopenia virus, another virus of the same family. This is a virus that infects not only cats, but minks and raccoons. And this canine parvo evolved from it, and the new virus no longer infects cats. So we have the original panleukopenia virus that infects cats, but the new one doesn't infect cats anymore. So it's changed its host range, and now it's infecting dogs. And the reason why it jumped into dogs is a very small amino acid change. And because this is relatively recently, this occurrence, this jump from cats to dogs, it was possible to get samples from cats and dogs all over the world and sequence them and figure out what went on. It turns out that the ancestor of this virus began infecting dogs in Europe as early as the early 70s, and then basically spread by fecal-oral contamination from dog to dog. And it was probably spread on the heels of many people. In fact, you step in feces on the street and you go to Europe and you bring the virus to Europe. And this has actually been shown in some cases to occur. Going from cat to dog requires only 2 amino acid changes in the viral capsid. Here's the capsid of the virus. It's an icosahedral shell. The receptor is the transferrin receptor on cells. Yes?
So if there's 2 amino acid changes, do you think that I guess the proofreading mechanisms in the cat cells somehow kind of pushed in that direction?
No, I don't think so. I think the virus existed as a minority population that was already able to bind.
And would it be beneficial for
You know, you don't know what direction you're going to go in, though, right?
But
So yeah, you could.
benefiting in a different direction would be beneficial.
Yeah, but it could also go in a lethal direction. So it's not a bad idea, actually, to take a chance and push it towards mutagenesis. But I don't think you could predict which way you would go. So I think what happened here is simply that there was a variant that multiplied very well in dogs, and it just dominated the population. As you'll see, that then after a few years it changed even further. It's starting to evolve with the dog population, and other mutations are occurring as well. So the virus binds this transferrin receptor, and there are the 2 amino acid changes allows the cat virus to now bind the dog receptor. So previously the cat virus only bound the cat receptor. Now the 2 amino acid changes lets it bind the dog receptor, and it no longer infects cats. And after a few years, that original variant is replaced by 2 new strains which have become more moderate in their ability to kill dogs. So it's an evolving host-pathogen interaction. So the principle here is that host strain switches by well-adapted viruses are rare. So the cat virus interaction was a stable interaction. Going to a dog is not very common, so it happened in this case. But when the switch occurs, it's severe for the new host, so the dogs died as a consequence. Once the dogs are infected, it's a new naive host. You have explosive variation and spread in the population. So this spread to dogs globally, basically. From one originating point, it spread to every dog in the world, potentially. Huge population because it was a totally naive population. So this was a really nice opportunity to study adaptation in the field. It was the first time this has really been done from start to finish. It's a great story. There is also a lot of emergence surrounding food animals. Last year's pandemic influenza coming from pigs is a great example. The pigs are raised for food. If we didn't have the pig industry, it's likely that that virus wouldn't have emerged. Another example is avian influenza, influenza viruses of birds. This in birds is a gastrointestinal infection. So in us, influenza is a respiratory infection. But in wild birds, it infects their gastrointestinal tracts, and that's a great way to spread the virus. You know, the birds are flying overhead, defecating, and they're spreading a nice sheen of influenza virus over the globe. The birds are fine. They don't get sick. It's a stable host pathogen or host virus interaction. In 1996, a variant of H5N1, avian influenza virus, emerged in China. It's not pathogenic originally in wildfowl, but it infected domestic poultry, chickens and turkeys and geese, became more virulent, and this is a virus that is now spread to domestic ducks and chickens, and it's becoming more virulent. It's spread globally now, and in 1997 it started infecting people. So whether this will spread among people is not yet clear. It's infected quite a few humans and caused a lot of death, but there is no convincing person-to-person spread. In less than 10 years, it's gone from China to all over the world. It's not yet in the Western Hemisphere. It's spread by migratory birds, so the virus of chickens that became more pathogenic went back into wild birds, and then, of course, they fly all over the place, and they spread the virus. But we also spread the virus because there's a huge illegal bird trade globally. People capture wild birds and send them all over the world. So we played a role in spreading the infection to Europe from Asia of this virus. Cats can be infected too. There was an incident a number of years ago in a zoo in Thailand. They were feeding tigers and these big cats chickens. Give them a dead chicken to eat. The chickens had H5N1 influenza. The cats got infected. So it can be spread to cats as well, and it can spread between the cats as well. So this is the current status of H5N1 in wild birds and in poultry and wild birds. All of Asia, pretty much Europe, parts of Africa, not yet in the Western Hemisphere. Seems only a matter of time before it gets here. But again, not yet convincingly transmitted among humans. As of the end of 2009, there were 467 human infections, 282 lethal ones, so that's a quite high lethality or fatality rate in these countries. And Indonesia has the highest number of infections. Vietnam and Egypt are up there as well. So again, these are mostly primary infections, dead-end infections from a bird to a human. As we mentioned with the noroviruses, some viruses emerge as technology allows. Rotaviruses, which we talked about as an example of acute infections, weren't discovered until we could figure out how to detect them. Recombinant DNA, PCR, lets you detect new viruses very readily. Hepatitis C virus was identified from the blood supply by DNA-based methods, and it was many years before it was possible to grow this virus in culture. And this is a hugely important liver pathogen, which was only picked up by new technology. We can detect and try and deal with emerging infections, but it costs a lot of money. So societies can deal with this, but you need money for research to figure out what's going on. You need a lot of money for early detection and identification technology. You need to have databases of everything that you sequence and make them available for everyone to look at, so if they identify new viruses, they can compare them. You have to have first responder actions. Every country now that's taken this seriously has a number of procedures built in, what to do in case there's an outbreak of an unusual disease. Public health action, vaccines, drug stockpiles, quarantines, all have to be worked out, and what's going to happen when you discover something new. Interagency communication, cooperation, a huge infrastructure has been built up now in government and academic and private institutions, all as a consequence of these novel viruses that have emerged. Now, I don't think we can predict what's going to come out, but what all this gives you is an ability to identify a new pathogen very quickly and to respond to it and perhaps contain its spread, as we saw really nicely with SARS. And that costs a lot of money in the end. So we can do a certain amount with emerging infections, but not without a cost.